Thanks a lot for the introduction, Robert, and thanks a lot, everybody, for hosting me today. I have a lot of interesting talks to many of you, and it's been an inspiring day for me. And it also made me realize that when choosing the title of my talk, I said, okay, understanding adaptive traits. This was, as in Dutch you would say, you put on too big trousers. You know, your trousers are too big. And this is because when talking to people here in this institute, I realized that people are really getting down to the detail of this. And this is in no way I'm going to go that deep. So if you look at my subtitle, it says Insights into the Genetic Basis of Sex Allocation in Nasonia parasitoid wasps. That's maybe more what I will be talking about. But it's all related. I first want to show you this slide, and this slide shows you a bit of the zoo that we are working on in my group. And these are all insects, as you can see. And here you see a predatory mite, which is attacking other mites. This is a parasitic wasp attacking Lepidoptera larvae. Drosophila suzuki, that's not a stranger to you. This is Nasidiocordis. It's a zoophytophagous bug, so it attacks plants and it attacks bugs. Here is a parasitic wasp called Trichogramma brassicae, and here is another pair. This is a parasitic wasp attacking an aphid. Inside the aphid, which you can't see, are endosymbionts, and endosymbionts can provide protection against these kind of attacks. So these are all the different systems that we currently work on, and they all can be, in this case, can all be applied to biocontrol, but they all have very interesting fundamental questions. But today I will talk to you about more fundamental work, which we did on sex allocation in Nasonia. And I must say that this work is already, it's not old, but it has a long history. So first of all, let's start with sex allocation. So what is sex allocation? So sex allocation can be defined as the investment of male versus female function. And in animals, it usually means the investment into sons or daughters in their offspring. And this has a direct effect on the fitness of a mother. It has been studied a lot, and there has been an interplay between theoretical work and empirical work, and it has been quite successful, as people are able to explain sex ratios in a wide range of organisms, ranging from tiny malaria parasites to these huge mammals such as red deer. Some people even call it one of the most successful areas in evolutionary biology. Then, if we talk about sex ratio of a population, you can look at it like this. You take a random sample of a population, which I did here, and then you determine the sex of all the members. And I did that using a handy tool called the website. So I just counted the number of males and females in your institute, and I came to 27 females and 22 males. Not sure if that's correct, but this serves as an example. The sex ratio then, and in this talk we'll be talking about the proportion of males in the population, is 22/49, so it's about 0.44. That's very close to 0.5, so very close to 50-50. So why would it be 50-50? Well, the explanation is given to us by Fisher, and those are called Fisherian sex ratios. And Fisher said in a large random mating population, 50-50 sex ratio is evolutionary stable strategy. It is like that because if you imagine that if you are a female laying eggs in this population, which is female biased, of course the best thing to do is to produce male biased offspring, because that gives you a fitness advantage. The other way around, if you happen to lay eggs in a more male biased population, the best thing to do is to produce more females. And like that you get stabilizing selection for a 50-50 sex ratio. And that's what we see in a lot of animals. However, it's not all 50-50. And it's of course very tricky to talk about the alpine marmot in a country with actual mountains coming from a lowland. But the alpine marmot has an excess of males, and that's because they have a cooperative breeding system, which favors males in this case. Here, this is an interesting animal. This a plover, a small cute bird. And here they reverse the sex roles. So the males actually take care of the offspring, whereas the females go out. And that's why you get an excess of males in these populations. Even more interesting, I think, are these. These are cleaning wrasses, so these fish that actually clean other fishes. And they live in groups of, say, 10 fishes. But there's only one male in those groups. So it's a very female by sex ratio in that. And it's interesting because if this male dies, then they're lost without a male. And then the biggest fish in that group, the biggest female, will turn into a male. And so on and so on. So it's very interesting. So this is not 50-50. Here's another example. This is the tiger snake in Australia. And that also has an excess of males. So it's not always 50-50. Another group that has weird sex ratios are parasitoid wasps. And parasitoid wasps are a big group. There are more than 50,000 species that belong to the Hymenoptera, so ants, bees, and wasps. Parasitoid wasps, females, they lay their eggs in the body or in the eggs of other insects. And these parasitoid larvae then eat and they consume the host, so the host insect. And instead of a host, a parasitoid will emerge again. And that's there are many different examples. This is a tiny one, again Trichogramma laying eggs on a butterfly egg. This is a huge one. This is called Pimpla, ovipositing inside larvae that live inside wood. So there's a huge variety. Because they kill their hosts, they are able to control the host population. That's why they are applied in biological control. And if you then work, and then I need to tell you a bit more about the sex determination, which is also interesting and special in parasitoids. So in general, females only mate once, and when they mate, they store the lifetime supply of sperm in the spermatheca. That's this tiny organ over here. And then each time a mother lays an egg, she can decide whether to fertilize an egg or not. And if she does that, she is actually determining the sex of her offspring. So here's the mother, and when she doesn't fertilize the egg, unfertilized egg will develop into a haploid male, whereas a fertilized egg will develop into diploid female. So this system is also known as haplodiploidy. So they have haplodiploid sex determination. Haplodiploidy means that the mother is also deciding on the sex of her offspring. And we have something called facultative sex allocation during oviposition. So that's interesting. And so what happens during oviposition? So first of all, a mother needs to find the host, and we skip over that completely. That's a whole ecological question. She then arrives at the host, and she starts judging the quality of the host. She does that by drumming on the host, tapping, drilling a hole in the host, and basically tasting the quality of the host. If she decides the host is good enough, she will inject venom in the host and start laying eggs in the host. And at that time, she decides on the number and the sex of the eggs. And she does that based on the quality of the host and on any social conditions, social interactions with other wasps. If you then want to work on parasitoids, Nasonia wasps are a good model system. They're actually the model organism for parasitoids, and they have been in the lab since the 1940s and probably before, but Nasonia research has a long history. Several interesting features is, of course, they have haploid genetics in a complex organism, but also there are about 4 interfertile species. So people use this to look at the genetics of traits differing between those species, like you have here, and you can hardly see that, but males of Nasonia vitripennis have tiny wings, whereas males of another species have longer wings. So that's the sort of stuff that people do. And as a consequence of that, there's a large set of genomic tools and resources that have been developed for this species. And one thing that people like to stress is that Nasonia has a full set of methylation genes, in contrast to Drosophila, and that's interesting. That's interesting too. So a consequence of this is that you find Nasonia in this environment, and that's of course the lab, where it's being studied by Nasonia biologists. Well, that's of course the lab side of things. This is where Nasonia occurs in the wild. This is a picture from the Hoge Veluwe. It's a natural reserve in the Netherlands. And it's this kind of forested areas where these wasps occur. And this is what their life cycle looks like. So Nasonia is a parasitoid of blowfly pupa. And these blowflies, they occur on carcasses, so on dead animals, not birds per se. And they also occur in nest boxes where these blowfly larvae suck the blood of young chicks. They then they lay eggs on these, they develop into larvae, these develop into pupa, and then into new flies. Unless a Nasonia female comes along and decides that the host is good enough, and then she starts laying eggs on the host, the Nasonia pupa, Nasonia larvae, and these can be 20 to 40 larvae per host. They then develop into pupa, and they emerge from the host. The males will emerge first, and they will wait on the host for their sisters to emerge, and then they will mate with their sisters. And then the males stay behind because they have only tiny wings, they can't fly, and it's the female that goes off and finds a new host, and then the whole cycle starts again. So that's the life cycle of Nasonia. It's important then to realize that they lay more eggs, more than one egg on a single fly pupa. Males are unable to fly, they compete for males, and one male can fertilize a lot of related females. And only the females, well, they mate on the host, and only the females disperse into new patches. And that leads to a situation where you have locally structured populations with patches like carcasses or nest boxes, and you get these locally structured populations. And if you have a situation like that, then the big question, of course, is in this case, what's the optimal sex ratio to produce? Is it still 50-50, or is it something else? Well, the answer is given by local mate competition theory. And that was thought of by Hamilton when he saw extremely female-biased sex ratios in insects and mites. And basically, this happens when related males compete for mates in a structured population. And in the case where you have a single foundress, you get very strong local mate competition. And that's here, because what happens, local mate competition, is when you produce more males, then these males will compete against each other for matings. So as a mother, it's best to optimize the number of males to that situation. And in this case, if you're the only female laying eggs, you should only produce so much males as to fertilize your own daughters. And there can be one male or can be two males, but not very much more. A very female by sex ratio. If there are more foundresses, and foundresses are then indeed other females laying eggs on a host, you have to produce more males. There's weaker local mate competition, and you get a more even sex ratio, up to the point where it gets about 50-50. Well, here you see what that would look like, and this is a graph from Nasonia. Here you see the prediction made by Hamilton. This is the model predicting the optimal sex ratio just based on the number of foundresses. And here you see if there are more foundresses, the sex ratio eventually gets to 50-50. That's the dotted line. And here you see different data points, and you see that the means actually follow this line quite well, although there's quite a bit of variation around that. And again, if you start looking into the wild, you see that there's variation. And there's actually quite a bit. There's variation. It's not, well, quite a bit. It's not too much, but it's significant. And it leads to a heritability of about 10%. And that's a broad sense heritability, so the difference between isofemale lines. The question, of course, is why is there variation? You can predict what's the optimal sex ratio, but they they don't, there's actually, well, variation around that. If you look into that, of course, you need to look at mutation, as mutation is the source of genetic variation. And we did a mutation accumulation experiment, and this was one of those old-fashioned mutation accumulation experiments where we didn't look what was going on in the genome. We just looked at the phenotype, where you set up replicate lines, you put them to many generations, and in the end, you look at the variance between a control line and your mutation accumulation line. You get graphs like this, control versus the mutated line, and from this you can calculate mutational parameters. One of those things is the mutational heritability of 0.001, and basically that says that there's one mutation per genome every 8 to 20 generations. Well, that's nice, but what does that mean? Well, actually, if you use these parameters to calculate the genetic variation that you should see in the field, you see that under a mutation selection balance, you would expect a higher genetic variance than you actually see. The logical conclusion from that is that the variation in sex ratio is actually being selected against by selection on other traits. So sex ratio is controlled by pleiotropic genes. It's not a very new finding. You also find that in other traits, but it's sort of a working hypothesis, looking at variation in sex ratio. So if we then look at sex allocation in Nasonia, we understand this pretty well based on models, but we don't know much about its genetic basis, and that's important to know if we want to understand the evolution of sex allocation. That's the fundamental or the basic side of things. And then for biological control, this can also be important, because in biocontrol, you use females to combat pest insects. And it's only females that you're interested in. So if you can increase the amount of females that you release, there can be a huge benefit. Let's see, that's sex allocation. First have a look at investigating genetic variation in Nasonia. And we did that, we wanted to have a tractable system in the lab. And in order to do that, we set up an outbred lab population. And to do that, we collected 5 field strains from the fields in the Hoge Veluwe in the Netherlands, forest area. And those field strains were basically everything you collect, one field strain is everything you collect from one nest box. That can be multiple founduses at the same time. We mix those and culture those to maintain genetic diversity. And that worked pretty well. We had a genetically diverse population with a pretty high effective population size, at least for the census size that we were able to maintain. That strain is used in selection studies, and for instance, Shu and Sha, who you have seen here during our internship internship of our PhD. She was here last year, I think, doing work. She worked with this population, doing genomic selection for wing size, and she's currently finishing up her PhD on that. So that's one population. Then we went a bit further and we tried to set up a genetic reference panel. And I know that when I say genetic reference panel to a whole, well, not a whole lecture hall full of Drosophila biologists, this is tricky, because you're all used to genetic reference panel of, say, 100 or 200 lines. And we managed to do something similar, but only with 34 lines. So that's not a lot. But the initial idea was to freeze the genetic diversity that was in our lab population, because the lab population, this HVRX, will deteriorate slowly. Then what we did is we set up mother-son matings. So we isolated virgin females from this population, and then they produce only males. So you can mate a mother to her own son. And then that was continued by 9 generations of SIP mating. So we had very much inbred lines that you can then store into diapause. So you can raise them under the right conditions, and you can just put them in the fridge, and they will stay good for about 1.5 years, and then you can take them out. So that's really nice. That's one of the benefits. You can then look at the intrapopulation genomic variation in those lines, and it's an analysis tool for complex traits. You can look at the heritability, or you can do GWAS studies. The nice thing, of course, about such a thing is that you can do repeated measurements of the same trait over and over again. So we then look at the molecular variation in this. We sequence the lines, not very deep, because this was done quite a while ago, and this is not up to current standards, but we sequence those 5 times per line. And if you look at the heterozygosity within the lines, we see that it's a very low heterozygosity within the individual lines. So the mean proportion of segregating SNPs per line is about 0.03, so 3% of the SNPs that are still present within the lines. It's not very high, actually lower than expected based on the amount of inbreeding that we did. If you then look at nucleotide diversity throughout the genome, it's 0.0012, and that is pretty similar to what we see in the outbred population that we keep in the lab. It's lower than what you see in natural populations, although this is a single gene estimate, and of course this varies throughout the genome. But the genetic diversity, the lines are a pretty good reflection of what we have in our lab population. And of course this varies throughout the genome, and here you see the nucleotide diversity plotted over the 5 different chromosomes of Nasonia, and you can see that it varies. It's slightly higher at the end of the chromosomes, lower in the middle where the centromere is. And this is a comparison. The colored lines is our reference panel, and the gray and black lines are the HVRX outbred lab population. Then of course you can calculate the GWAS on that, and if you do that, you see that it's much lower in our inbred line panel. And that's not so strange, because of course we've been doing strong inbreeding followed by expansion of those lines. So you see a negative to GWAS here. Looking at the linkage equilibrium, there's pretty strong linkage here, and that's again probably due to inbreeding. You can see that the half decay distance of the linkage in this population is about 18 kb, and that's pretty large if you compare it to your favorite animal, Drosophila, which is very small, and of course it has a huge population size. The honeybee is even smaller, but honeybee has a very high recombination, which lowers this distance. Of course, in this case, it's most likely due to the population structure that we've created. Although it must be said that Nasonia, by nature, is a natural inbreeder, so that can be that can also contribute. Although we need to look more into details to get a proper estimate in wild populations as well. And then the extent of LD varies within the chromosomes, and you can find very large linkage blocks ranging from 10 to 36 MB. So that's again a lot of inbreeding, but indeed, but you see variation over those chromosomes. If you then look at the phenotypic variation, and I will get to the sex ratio a bit in a bit. Here are estimates for some different traits. So this is the variation found in head width between the different lines, wing length, and mean starvation resistance. You see variation, and that results in significant heritability between the lines. So for those traits, and here are a few other estimates. So this is longevity. That's again longevity, and here is the total lifetime reproduction. Again, there is strong heritabilities there. So it's interesting to look at the genetic basis of that, which I won't go into right now, because I want to discuss the variation in sex ratio with you. So, sex ratio variation. We started looking at that, but then we realized all these lines, when we started off, had a Wolbachia infection. And in this case, Wolbachia infection causes a cytoplasmic incompatibility. So if one strain has Wolbachia and the other hasn't, or they have a different type of Wolbachia, the cross is incompatible. So in this case, we tested only for Wolbachia infected lines, and those were 26 of our lines. And we tested a single founder's sex ratio of about 40 females per line. And the phenotyping is pretty easy because you just count and you sex your offspring. And then you get 2 phenotypes for the price of 1. So you get the sex ratio and the clutch size. And we did that in a lot of broods. And here you see the distribution of those. You see immediately that the sex ratio is very much skewed towards the female side. So this is the proportion males. And here the clutch size is pretty normally distributed. And this is a plot of the variation. And you see that there's quite a bit of variation, but you see that there's, at least this is a strong outlier, line number 62. And we discovered that it has a transient Wolbachia infection. So the Wolbachia infection was actually going out of this line. And that's reflected in a high sex ratio and a low clutch size. That's because you get incompatible crossings, resulting in the female zygotes to die. That results in a low clutch size, but a high sex ratio. So we took this one out. And if you do that, you get to heritability for sex ratio of about 10% and clutch size about 8%. And we decided to do a GWAS on those lines. And I have a picture here from Joost van der Heuvel, who some of you might know. He helped me out with statistics on this. But basically, and these are the details, for a sex ratio, as it's a binomial distributed trait, we fitted a generalized linear mixed model on that. And we did single marker regressions there, with line fitted as a random variable. We did the same, but then a linear mixed model for clutch size. And then we determined the significance thresholds by permutation. For sex, we tried to go for a Q value of 0.1, and for sex ratio that meant that we had a threshold of 5.7. For clutch size, we didn't even get there, because if we have a Q value of 0.1, the minimum Q value here was 0.46, so no variant was significant. So we couldn't use this for determining significant peaks. Finally, I said from our, we were also interested in finding a shared genetic basis, shared genetic background between sex ratio and clutch size. And we solved that in this case by looking for an overlap in the test statistics between the 2 traits. So looking at the p-values in 400 kb windows in real data versus a permutated data set. So this is what it looks like. If you then do the GWAS, you get a single peak passing the thresholds on chromosome 1. Still, you see a lot of peaks, so it's evident or indicating that it's a polygenic trait. And all those SNPs in this peak are in high linkage with each other. So they also give the same signal, basically. And those are 18 SNPs that are significantly associated with sex ratio. The nice thing, though, is that those are common variants, so it's not just one line that has that has this causative SNP, but it has an average minimal allele frequency of 0.42. And then, of course, you can look at what's behind there. Well, we find those 18 SNPs. The majority are intron variants and upstream variants, downstream variants, but there are 2 of them that are related to interesting candidate genes. So one of them is a 5' UTR variant in ADP ribosylation factor-like protein 4C. I get to the details in the next slide. And the other one is splice region variant in cilia and flagella associated protein number 52. Well, what does that mean? So the first one, it's an ADP ribosylation factor, ARL4C, and it's involved in the regulation of vesicular trafficking. And there's not much known about this one. There's a related ARL4A that's strongly expressed in the testes of mice. And interestingly enough, a disruption of this protein results in reduced sperm counts. So it might has something to do with reduced fertilization or anything. However, in our lines, this variant didn't cause a premature start codon, and the role of this ARL4C is still unclear. It's more interesting to look at the other variant, and that's a splice region variant in this cilia and flagella associated protein. That's a highly associated protein, or highly conserved, protein, And the human ortholog of this, it's called WDR16, it occurs in the sperm tails of humans. And in low motility sperm, we actually see a low expression of this gene. Interestingly, it's highly expressed in Nasonia testis, so it likely has something to do with Nasonia sperm as well. It means it's a splice region variant that could affect the functioning of this protein. And it means that we can have a direct effect on sex ratio possible through the sperm. So actually it means that if the sperm is not functioning well, it's stored here in the spermatheca, then the mother can try to fertilize the egg. But of course, if she fertilizes it with non-functioning sperm, not much will happen, and then fertilized eggs will actually develop into males instead of females. So here we are on to a male effect on sex allocation, and that has been observed before in Nasonia in this study, where they looked at different males mating to the same female strain, and there's tiny but significant changes between strains. So there's a tiny effect of males on the sex allocation as well. That's interesting because we actually tend to think that sex allocation is a female business, but males have an effect here as well. Then finally, if we look at the overlap between sex ratio and clutch size, we find evidence for that on chromosome 5. So here are two regions that show overlap. We didn't go into the details of that. It's important to stress that there's a shared genetic background. It's a bit too early to really dive into what genes are under that. But finding a shared genetic background actually matches the prediction made by these mutational parameters. So genetic variation for sex ratio is restrained by selection on pleiotropic loci affecting other fitness traits. Well, of course, it's here we're looking at clutch size. You can say, well, yeah, fitness traits. Why look at clutch size? It's just a trait that you get for free with your phenotyping. That's of course true. You could look at any fitness trait, but there's actually a prediction from a theory that these two are correlated. So, as I told you before, a single mother should only produce enough sons to fertilize all of her own daughters, and not anymore, because then she's wasting resources. It means that if the clutch size increases, you should see an increase in the number of. Now I'm getting confused. Now it's getting interesting. So you can wake up all. If you see the clutch size increasing, you should see an increase of the number of males. And what we found was actually the other way around. You see a weak correlation between sex ratio and clutch size. Clutch size increases, and you see less males. It's only a weak correlation, but there is at least a correlation between those two. In other studies, where they looked at this in Nasonia, it is the other way around. In our data, it's just not strong enough. But it's an interesting observation here. So, if you look at this panel of lines, we found substantial variation for sex allocation. It's sort of the first genomic visualization of sex ratio in Nasonia, and we show polygenic basis, overlapping genetic background with clutch size. And we found some interesting candidate genes, I think, which points to a role for males in sex ratio variation. And of course, this is only correlational evidence, and we need a follow-up. And we're now working on the Splice variant analysis. And after that, we tried to get a knockdown. RNAi works really well. However, knockout, so CRISPR-Cas9, really doesn't work well yet in Nasonia. So it might work in other species, but in Nasonia, basically, if you try to inject the embryos, they're really tiny, as they are in Drosophila, and they just blow up. And people haven't solved this problem yet. It seems to be really down to technical details of properly doing this. And of course, the question, is this present in newly wild-caught lines? Because I've been talking about a lab population and not about a wild population. So conclusions from this, okay, yeah, we only have 34 lines. That's not a lot, but we find significant heritabilities for a wide range of traits. You could argue we need further extension of this, this genetic reference panel, and it's nice because it allows the measurement of repeated measurement of phenotypes. However, you could also question whether you need these reference panels at all. We might say, okay, yeah, we add lines to this, but do we really need them? Because the time we did this was about 5 years ago. Then it was a very good idea to do this. Now, of course, sequencing is cheaper, and there are other techniques. So you could do rot-seq or something else. So you could directly study these animals in the wild. Although I must say that one downside to studying Nasonia as compared to studying, for instance, Drosophila, if you put out a bucket of rotting bananas, you can capture a lot of the genetic variation in Drosophila, because you can just collect the flies. In Nasonia, if you the way to collect them, the easiest way is to empty nest boxes, but you never know whether that's due to one founding female or to many founding females. So as of now, it's been really difficult to collect females directly from the wild. And in collaboration with another lab, we're working on getting a proper trap for this, which actually contains rotting animals. So that's not a nice story, which I'd rather skip right now. Or maybe it's interesting. But if you want to know more, you can ask me about that. With that, I quickly like to acknowledge the following people. The ladies here on the top. So Gabriella, Berta, and Jade, they did a lot of the work, the molecular work and the wasp pushing work. Then this work is a collaboration with Nikki Cook, Joost van der Heuvel, Louis van der Zanden, and Dave Schuker, and it's been funded by NERC in the UK and the Netherlands Genomics Initiative. And with that, I'd like to end with this picture and with thanking you for your attention and inviting your questions.